If it's very, very dense, it moves farther down through the salty solution into a very, very salty area. If it's less dense, it doesn't move very far at all. So when you spin one of these tubes in a centrifuge, you use a centrifuge that kind of looks like this, where it's called a hanging bucket. And these buckets, as it spins around the center, it'll rotate like this. The buckets will actually rotate up on the hinge, kind of like, I'm going to demonstrate a centrifuge right now. You guys ready for this? Just kidding. So what I'm going to do is I'm going to spin this around, and as I do, you're going to see that it starts off hanging down like a bucket, but as I spin, it's going to start going up until it's totally horizontal. You guys see that? And now when I release these other paper clips, hopefully you'll see them move down towards the bottom. You guys see that? That's what a centrifuge does. So when you put one of these buckets, you fill it, and you spin it, it rotates up, and then whatever was on top moves directly down toward the point where it's at the same density as the solution around it. So the cool thing about this is you can separate molecules based on density. And that's the key to this experiment. Okay, so you guys good with centrifuges? And these centrifuges, by the way, they're really, really powerful centrifuges. They spin, like 100,000 times G. So they might be rotating at 50, 60,000 RPMs, like really, really, really fast. They're super dangerous. They have these, some of them are like the size of huge washing machines and have like steel plating on the outside, so if something goes wrong, you're protected from a projectile coming through and killing you. And usually that works. So, usually, not always. So the only other thing I need to tell you about is, or remind you of, are isotopes. And we talked about isotopes last time. You guys remember isotopes? So what's an isotope? It's a version of the same element that has different numbers of neutrons. And remember, neutrons confer mass to an element. And so if you add additional neutrons in, you're going to make a denser atom. And if you build a molecule out of those atoms, you're going to make a denser molecule. So what we're going to use is either normal nitrogen, which is nitrogen 14. So it has 7 protons and 7 neutrons. Or we're going to use heavy nitrogen, which is nitrogen 15. All right. So imagine, if you will, for a moment, that ignore everything except the far left side of this for now. We're going to grow some bacteria in a culture where we feed them just regular food with regular nitrogen 14. And to help you with that visualization, I built a petri dish as the universal symbol for bacterial growth. So imagine bacteria growing in a normal nitrogen medium. If we let them grow forever, take some out, extract their DNA like you guys did on Monday, right? Load it on top of one of these tubes, and then spin it in a centrifuge. Well, that DNA you extracted is going to migrate down through that salt to the point that it has sort of an equal density to the solution around it. Have that in your mind? This is where all the DNA migrates for bacteria growing with normal nitrogen. Now look over to the next category here. We've got bacteria growing in a heavy nitrogen medium. That means that all of the DNA of that molecule, because DNA is rich in nitrogen, right? You guys remember that? Do you remember the name of the part of the molecule that is the A, the T, the C, or the G? Nitrogenous base, which just means it's nitrogen rich. So there's tons and tons of nitrogen. So if you grow bacteria in a heavy nitrogen medium, extract the DNA from it, load it on top of one of these tubes, and then centrifuge it, it'll move down through that gradient to a lower point because it's denser. Because every nitrogen has been replaced with a heavy nitrogen version. Okay, that's the setup for the experiment. Now this is what they did. They took bacteria that had been growing forever, I mean like thousands of generations in a heavy nitrogen medium, and they shifted it to a low weight, the normal nitrogen medium, for 20 minutes, which is about how long it takes for the bacteria to synthesize a whole new copy of all their DNA and divide. So 20 minutes in this case is sort of the magic number for completing or copying all the information and dividing. So you then take the DNA from it, you load it onto one of these tubes, you centrifuge it, and the DNA all migrates to this point in the tube. Okay, so that's our first bit of data. The second bit of data is we let it go for 40 minutes, which means it's gone through 2 rounds of DNA copying, formerly in a heavy nitrogen medium, now 2 rounds in light nitrogen, and we end up with DNA bands at these 2 points. I want you guys to discuss with your group what type of replication this data supports. Okay?